Okay, so welcome. This is the start of Lab 7. This is a photosynthesis versus respiration lab, and I'm going to show you how to set it up. You're going to have trays just like this that Rosemary or Kapoor are going to set up. And the first part of the experiment is going to be looking at the light reaction versus the dark reaction. So what students are going to do is they're going to take 4 of these test tubes. I've grabbed 2. They're going to fill it up about 2/3 of the way with water. They're going to go ahead and add 15 drops of phenol red, and it'll have a really deep red color. They're then going to go ahead and take a straw. There'll be some straws in the back. And they're going to mix up the tubes and blow in the tubes till the phenol red go from a really dark red color to like a orangey color. And what the phenol red is, it's a pH indicator, and by adding the CO2, it's changing the pH. So we want to get them to at least have all 4 of the tubes match in color. So that might take a little time. So they're going to have 4 of these tubes, 2/3 of the way with water, 15 drops of phenol red, and they're mixing up the phenol red and blowing in it until all the colors come out to like an orangey color. Then what they're going to do is they're going to take about a 5 to 6 inch piece of Elodia and put it so the cut side end is up in one of the tubes. So here I have 2 sets of tubes. One is going to be just with water with my phenol red in it. This one is going to have my phenol red, water, and an Elodia tube. Let's call this my light reaction. What's going to happen is these tubes will be put in this setup here, and it'll be set up perfectly just like this for you. They're going to put these tubes in the water and let it sit for about an hour and a half. The other 2 tubes that they're going to set up that are going to be like this, again, is going to be one control one and one with the Elodia, and that's going to be put in this little beaker, and under their desks they have one cupboard with a sink attached to it. They're going to put that there. That's going to be the dark reaction. So what's going to happen is those tubes and these tubes will sit for an hour and a half, and after an hour and a half we'll see a pH change, and they're going to have to talk about that. So the first thing that you're going to have to make sure is that they only put their tubes in this one row, and then they mark the tubes in a way that they can recognize that those are their tubes. All right, I think we're good.